everybody. Welcome to Module 9.1. This is the first module in Module 9 and the second set of modules in Unit 4. Like I told you about in the last module, Module 8, this one's set up in the same way. So this first one is going to be an overview of everything that we need to know. And then the subsequent three videos will be, as well as PowerPoints, will be more in-depth information about these topics. So over here, we're just going to start with a little bit of sensory integration. And what you can see here, sensations are detected and carried into the brain. Everyone has the same sensation. You'll see the same thing. Sensory receptors are stimulated. And then all types are electrical signals and include generally an action potential. And then perceptions are constructed by the brain, or what you see based off of that sensation or that electrical impulse. And so we'll perceive that sensation or that stimulus differently. And that'll be integrated with that sensory input, and then multiple brain region communication based off of your individual experiences and what actually makes up you. So here we're going to get into some of the types of sensory receptors and how they're classified, and we're going to talk structurally here. We have free nerve endings, which are dendrites embedded in tissues that would receive a sensation. For example, pain and temperature receptors in the dermis of the skin. You can have encapsulated endings, which are sensory nerve endings that are encapsulated in connective tissue that enhance their sensitivity. Examples of those include the lamellate corpuscles that respond to pressure and touch that are embedded in the dermis. And we also have receptor cells, which has a distinct structural component that will interpret a specific type of stimulus, for example, photoreceptors in the retina. We can also classify them based off their location to the stimulus. We can have exteroreceptors, so a receptor that is located near a stimulus in the external environment, such as the somatosensory receptor in the skin. The interoreceptor, so those interpret stimulus from internal organs and tissues, such as the receptors that sense increases in blood pressure in the aorta. And the proprioceptors, so receptors that are located near a moving part of the body, such as a muscle, that interpret the position of the tissues as they move. Also talk about some of the functional types. So we can have chemoreceptors, which interpret a chemical stimulus. Osmoreceptors, which respond to solute concentrations. You have nociceptors, which detect pain. Mechanoreceptors, so it'll interpret a physical stimulus. And then thermoreceptor, which are sensitive to temperatures. We also have some sensory adaptations, so that's diminished sensitivity as a consequence of constant stimulation. For example, what we consider to be quote unquote high pain tolerance. All right, getting into some of the senses themselves. We have gustation or taste, which provides information about the foods and liquids we consume. Some of the basic anatomy of taste, although for sensation and taste can be distinguished, there appears to be really no difference in the structure of taste buds, and taste buds in all portions of the tongue provide all four basic taste sensations. So four classic sensations of taste that we have, and two new ones that have been identified relatively recently. So these are sweet, which can be stimulated by sugars, alcohols, saccharin, and some other amino acids. Salty, stimulated by sodium chloride. Sour, stimulated by acid. And we're almost about 1,000 times more sensitive to sour than either sweet or salty chemicals. It tastes bitter, which can be stimulated by alkaloids that are commonly found in bitter tasting plants like tobacco and coffee. We have umami, which is a relatively new discovery that's identified by the Japanese, and it appears to be responsible for the beef taste of steak, or the twang of aging cheese, the flavor of the food additives such as like MSG. And that can only be detected by the circumvallate papillae. We can also have fats and lipids, which there is new evidence that recently suggests that this may also be newly discovered taste sensations. Humans also possess water receptors, and they're especially concentrated in the taste buds located in the pharynx. And these sensory receptors signal the hypothalamus to help maintain some of the water balances. Some of the taste receptor cells, or gustatory receptors, are distributed over the superficial or the superior surface of the tongue, as well as projections called lingual papillae, and the adjacent portions of the pharynx and the larynx. So the human tongue possesses four primary types of lingual papillae. You have the circumvallate papillae, which are 7 to 12 relatively large round papillae that are shaped like the tip of a pencil eraser that are surrounded by like deep epithelial folds. And these are found on the posterior margin of the tongue in kind of like a V pattern. And each circumvallate papillae possesses about 100 taste buds along the sides of them. You have the fungiform papillae, which are mushroom shaped papillae within the shallow depression scattered along the anterior two-thirds of the tongue. And these typically possess only about 5 taste buds located near the top. You have the filiform papillae, which are hair-like papillae scattered all around the tongue, which don't have taste buds associated with them. And these are designed to create friction on the surface. And then you have the foliate papillae, which are found on the posterior and lateral edges of the tongue and look similar to the pages in a book. So many of the papillae contain taste receptors or test receptors and specialized epithelial cells and sensory structures called taste buds. And although we have about 10,000 taste buds when we're young, an adult only possesses about 5,000 taste buds by age 50. So about less than a third of the taste buds still exist. And taste buds contain 3 types of cells. You have the gustatory receptor cells, which are epithelial cells that extend numerous slender microvilli called gustatory hairs into the surrounding fluids through the taste pore, which is a narrow opening at the surface of that bud. And each taste bud possesses about 40 to 100 gustatory receptor cells. However, each receptor cell lives only about 10 days before it's replaced. And these receptor cells are sensitive to the chemicals contained within foods that are ingested and release neurotransmitters based on the amount of chemicals in the food. Neurotransmitters from the gustatory cells can activate sensory neurons in the facial, the glossopharyngeal, and the vagus cranial nerves. We can also have supporting cells, which form an insulating capsule and make up the bulk of the taste buds. And then we have the basal cells, which are the gustatory stem cells that divide by mitosis to replace some of the dead or injured taste buds. So getting into some of the physiology of taste here. So in order for a chemical to be tasted, it must dissolve in saliva, diffuse into the taste pore, and contact the gustatory hairs. When the chemicals contact the gustatory hairs, depolarization of that cell membrane is triggered and an impulse is into the brain. So salt and sour receptors are chemically gated sodium channels, and the stimulation of that produces a depolarization by the influx of sodium into the cell, and depolarization of the membrane stimulates the release of neurotransmitters. When things like sweet, bitter, and umami bind to the membrane receptors, and an active G protein called the gustactins is activated and it produces a second messenger on the inside of that membrane. And activation of the second messenger stimulates the release of the neurotransmitter. So the taste sense is served by 3 cranial nerves: nerve 7, nerve 9, and nerve 10. So the facial nerve, nerve 7, innervates the anterior two-thirds of the tongue, and the glossopharyngeal nerve, nerve 9, innervates the circumvallate papillae and the posterior one-third of the tongue, and the vagus nerve innervates the taste buds scattered along the pharynx of the epiglottis. So all 3 nerves send impulses through the solitary nucleus of the medulla oblongata, and from there the impulses are sent from the thalamus and the taste cortex within the insula of the cerebrum. And taste is about 80% smell. So when olfactory receptors are blocked, food tastes rather bland. And although it's not considered gustation, the trigeminal nerve, or nerve 5, is able to relay information regarding the texture and the temperature. So speaking for food to be relatively spicy in nature would be determined by that. Moving into olfaction or smell. So the basic anatomy of that, the sense of smell or olfaction is provided by pairs of olfactory organs. And these organs are located in the nasal cavity on either side of the nasal septum. So the olfactory organs are composed of 2 layers. We have the lamina propria, or the layer of areolar connective tissue containing numerous blood vessels and nerves. And this layer also contains the olfactory glands, or the Bowman glands, whose secretions absorb water and form a thick pemmican mucus. Olfactory epithelium, which are layers of cells that are sensitive to odorants, dissolve chemicals that bind to plasma proteins found on the surface of olfactory receptor cells. Olfactory epithelium is composed of 3 kinds of cells. So we have olfactory receptor cells. So these are bipolar neurons whose exposed tip form a knob that projects beyond the epithelial surface, and the knob provides a base for up to 20 cilia that extend in the surrounding mucus and lie parallel to the epithelial surface. And at least 10 to 20 million olfactory receptor cells lie in the nasal epithelium. This area is only about 5 centimeters or so. And olfactory neurons are regularly replaced within the nasal epithelium, after which the axons of the new neurons kind of have to find their appropriate connections in the olfactory bulb. We also have supporting cells, which are columnar epithelial cells that insulate the olfactory cells from each other. And then lastly, the basal cells. So the olfactory stem cells that lie between the supporting cells, and these divide by mitosis again and differentiate into those olfactory receptor cells. And that rapid turnover is one of the few examples of neuronal replacement in adult humans. So the axons of the olfactory receptor cell collect in about 20 or more bundles that penetrate the cribriform plate of the ethmoid and constitute the filaments of the olfactory nerve that synapse within the olfactory bulb. The olfactory bulbs then transmit the impulse towards the brain via the olfactory tracts to reach the olfactory cortex within the temporal lobe of the cerebrum, the hypothalamus, and portions of the limbic system. Some of the physiology of smell. So detection of primary sense requires the odorant be dissolved in a liquid. So that usually is nasal mucus. Odorants bind to plasma membrane proteins, which then lead to the activation of the adenylyze cyclase and the enzyme that converts ATP to cyclic AMP, which is cyclic AMP. So then cyclic AMP opens sodium channels in the plasma membrane and as a result begins to depolarize. And if sufficient depolarization occurs, the action potential is triggered and the axon and the information is relayed to the CNS. Recognition of many senses due to patterns of activity in the brain that arise from activation of different combinations of olfactory receptors. And the olfactory cortex is located within the medial aspect of the temporal lobe. So loss of your sense of smell is due to damage to the olfactory nerve, and that's called anosmia. Humans also have a pathway that's associated with the limbic system, and from the thalamus to the amygdala and then to the hypothalamus, and that's involved in the perception of emotions and may explain why some scents can endure longer and incur emotions in us. Moving on to the ear, so some of the basic anatomy. So the external ear, which is the visible portion of the ear. You have the auricle, which is the external projection composed of elastic cartilage and adipose tissue, and that surrounds the opening to the ear and collects sound waves into the external auditory medius. So the external auditory medius, so a short curved tube that extends from the auricle into the eardrum and funnels sound waves towards the middle ear. And the skin lining this passage contains numerous ceremonous glands that secrete the waxy cerumen and possess many small outwardly projecting hairs. So what essentially makes the ear wax. Together, the hairs and cerumen help to keep foreign objects and insects from reaching the more delicate internal structures and also reduces the growth of microorganisms. We have the middle ear, which is an air-filled cavity, also called the tympanic cavity, which is separated from the external auditory meatus by the tympanic membrane. So the tympanic membrane, also called the tympanum or the eardrum, is a thin, semi-transparent sheet that vibrates to amplify sound waves. We also have the auditory tube. And this tube is normally closed, but during swallowing and yawning will help to equalize pressure in the middle ear cavity. Unfortunately, it can also allow microorganisms to travel from the nasopharynx into the middle ear, and that can result in middle ear infections known as otitis media. We also have auditory ossicles, and the three smallest bones in the body that help to amplify the sound. So it's technically 6 total bones, 3 in each ear. We have the malleus or the hammer, and the handle is secured into the internal surface of the eardrum at 3 separate points. We have the incus, which can be considered the anvil, which attaches to the malleus and to the innermost stapes. And then the stapes were the stirrup, and that's bound to the edges of the oval window, which is an open end of the bone that surrounds the inner ear structures. And immediately below that oval window sits the round window. And while the oval window transmits sound waves through the inner ear, this opening allows sound waves to dissipate. Talk about some of the muscles in the middle ear. We have the tensor tympani muscle, which is a short ribbon of muscles whose origin is in the petrous portion of the temporal bone of the auditory tube. And when this muscle contracts, it tenses the eardrum by pulling on it medially to prevent damage from very loud sounds. And this muscle is innervated by the trigeminal nerve. We also have the stapedius muscle, which is innervated by the facial nerves. And it checks the vibration of the whole ossicle chain and limits some of the movement of the stapes. So some of the anatomy of the inner ear, that contains some of the sensory organs for hearing and the equilibrium in the outer bony labyrinth that's filled with perilymph, and intermembranous labyrinth filled with endolymph. So the bony labyrinth is a shell of dense bone that surrounds and protects the membranous labyrinth. And within the bony labyrinth flows perilymph, which is a liquid whose properties closely resemble that of cerebrospinal fluid. The bony labyrinth is composed of three parts. You have the vestibule. Um, so it's a central egg-shaped cavity that houses some of the receptors for static equilibrium or linear movements such as acceleration, deceleration, and head position relative to gravity. You have the semicircular canals, and these are three canals that contain the receptors for dynamic equilibrium or the rotational movements of the head. And you have the cochlea, which is a snail-shaped structure that contains the receptors for hearing. Moving on to the membranous labyrinth, which is a collection of fluid-filled chambers that contain endolymph rather than perilymph. And the vestibule contains a pair of membranous sacs that are filled with endolymph. So you have the utricle, which is a slightly larger duct located on the top of the vestibule, and the saccule, which is a small duct that's located on the bottom of the vestibule. The cochlea is composed of three chambers, but only one part of the membranous labyrinth. You have the scala media, which is a slender, elongated middle chamber, also known as the cochlear duct. And the cochlear duct is filled with endolymph and houses the organs of the chordate. The organs of the chordate are composed of mechanoreceptors called hair cells and a tectoral membrane. The cochlear duct sits kind of sandwiched between pairs of perilymph-filled chambers that are called the scala vestibula and the scala tympani. So the scala vestibula, which is the upper chamber, also known as the vestibular duct, and its floor forms the vestibular membrane. And then the scala tympani, which is the lower chamber, also known as the tympanic duct, whose ceiling kind of forms the basilar membrane. And there are three semicircular ducts that are located within the semicircular canals. You have an anterior, posterior, and a lateral duct. And these receptors within the semicircular duct provide sensations for the rotation of the head. You can speak a little bit about the vestibular cochlear nerve here. And cranial nerve 8, the vestibular branch transmits impulses regarding some of the balance and equilibrium from the vestibulae and the semicircular canal to the CNS. And the cochlear branch relays impulses regarding hearing to the CNS. This is a little bit more about the cochlea here. A little bit about transmission of sound waves to the cochlea. So a sound wave that causes the tympanic membrane to vibrate, and this vibration is amplified as it moves across that the malleus, the incus, and the stapes. And the amplified vibration is picked up by the oval window, which again causes those pressure waves in the fluid of the scala vestibuli and the scala tympani. And the complexity of that pressure wave is determined by changes in amplitude and frequency and the sound that enters the ear. So talking a little bit about the physiology or the pathway, the pinna directs sound waves to the external auditory medius, and the tympanic membrane vibrates according to high-low frequencies. Vibration is then again transmitted from the eardrum to the malleus, and then to the incus, finally to the stapes. And all these structures amplify that sound wave. The stapes vibrates and then pushes that membrane to the oval window in and out. Movement to the oval window then cause fluid pressure in the perilymph of the scala vestibula, and those pressure waves travel through the cochlea towards the round window, and in doing so, kind of depress and deform the walls of the scala vestibula and the scala tympani, and that collectively causes the basilar membrane to move. So that movement or the vibrations in the vestibular membrane cause the basilar membrane cause the hair cells to move against the tectoral membrane. And the bending in the hair cells against the tectoral membrane depolarizes the hair cells and generates a nerve impulse that travels to the CNS via the vestibular cochlear nerve. Nerve impulses in hearing are interpreted by the auditory cortex, which is located in the temporal lobe of the cerebrum. Pitch is determined by the frequency of the sound wave and is measured in hertz. And so you can have high frequencies, which are high pitch, and low frequencies, which are low pitched. Volume is determined by the amplitude of the sound wave and is measured in decibels. So high amplitude equals a low sound or a loud sound, and low amplitude equals a soft sound. We have a few disorders of the ear. One we already talked about, otitis media, which is an infection in the middle ear. You can have otitis externa, which is an infection of the external auditory canal. You can have otalgia, which is an earache. You can have presbycusis, which is the loss of the ability to hear high-pitched sounds. You can have deafness, which is any hearing loss in general. You can have conduction deafness, which is something that hampers sound conductions. So such as too much earwax or something like that. And you have sensorineural deafness, which results from damage to the neural structures at any point from the cochlear hair cells and to including the auditory cochlear cells. You also have vertigo, which is loss of balance and equilibrium due to inner ear problems that can cause motion sickness, headaches, nausea, flushing, sweating, and vomiting. And then you can have tinnitus, which is a ringing or clicking sound in the ears. This essentially highlights just the end portion of what we just talked about. So, um, basically, the hair cells will stimulate the vestibulocochlear nerve, and that travels along all the way till it gets to the primary auditory cortex, where that'll be interpreted in the brain. So, speaking of the physiology of equilibrium here, the equilibrium receptor regions in the inner ear are called vestibular complex, and impulses from the vestibular cortex are sent via the vestibular nerve fibers to the brainstem and the cerebellum. These areas then coordinate visual stimuli with the active muscle contractions. And there are two types of equilibrium. You have static equilibrium, which is helps to maintenance of the position of the body relative to the force of gravity, um, and is detected by the scalae and the utricle of the vestibule. So the hair cells of the scalae and the utricles are clustered in oval structures called the maculae. And the maculae consist of hair cells that possess about 80 to 100 stereocilia, um, and a single kinocilium. So the stereocilia and the kinocilium are embedded in an overlying gelatinous mass that contains some densely packed calcium carbonate crystals known as statoconia. And the whole complex is called the otolithic membrane. So linear movements cause that otolithic membrane to move and pull on the hair cells. So when your head is in the normal upright position, the statoconia sits atop the macula, and the weight presses on the macular surface, which pushes the hair cells um, process down rather than to one side of the, or another. So when your head's tilted, the pull of gravity on the statoconia shifts them to one side, and that thereby distorts the hair cell processes, which stimulates that macular receptor. Um, a similar mechanism kind of accounts for the perception of linear acceleration when your car speeds up suddenly. So the statoconia lag behind, and the effect on the hair cell is comparable to tilting your head back. We also have dynamic equilibrium, um, which talks about some of the maintenance of body position, specifically the head in response to rotation. So the receptors for the dynamic equilibrium are within the ampulla, which is found at the base of the anterior, posterior, and lateral semicircular ducts. So the ampulla consists of a tuft of hair cells known as cristae, whose microvilli are embedded in the gelatinous cupula. And when the head rotates in the plane of a semicircular duct, the movement of the endolimbs along the length of the duct pushes the capilla to one side, which distorts that receptor process. Moving the fluid in one direction stimulates the hair cells, and moving in the opposite direction inhibits them. So when the endolimb stops moving, the elastic cupula rebounds to its normal position. Each semicircular duct responds to one of the rotational movements. So a horizontal rotation, as in shaking the head no, stimulates the hair cells of the lateral semicircular ducts, and nodding the head yes excites the anterior semicircular ducts, and tilting your head to the side activates receptors in the posterior ducts. All of these can contribute to motion sickness as well. This just again highlights linear acceleration as well as rotational coding. All right, moving on to the eye. So some of the basic anatomy there. So the adult eye is a sphere with a diameter that has about 2.5 centimeters, and contains a lot of different structures of the eyeball. Before we get into a lot of things here, I want to talk about some of the disorders that are associated with the eye. So you have myopia, or also called nearsightedness. So in that disorder, a person can see objects, close objects, but can't see distant objects. They're kind of blurred. That typically results from the eyeball being too long, so that the focal plane is extended and the light diverges again. Corrective lenses have usually one concave surface in that instance. You can have hyperopia, or also called farsightedness, and which is basically the opposite of that. Um, you have an astigmatism, which is an unequal curvature of the lens of the cornea. You have cataracts, which are clouding of the lens that cause the world to appear kind of distorted. Glaucoma is increased intraocular pressure, which may increase the dangerous levels and compress the retina and optic nerve, and usually occurs if the draining or the aqueous humor is blocked. You have retinal detachment, in which the retinal detaches from the vascular layers and allows like the jelly-like vitreous humor to seep between them and can cause permanent blindness. Um, you have diplopia or double vision, strabismus or cross-eyed. You can have color blindness, which is a congenital lack of one or more of the cone types. It's usually inherited as a sex-linked condition and is far more common in males than in females. Um, and you can also have nycloptia, which is night blindness, and is usually a result of a vitamin A deficiency. Um, this won't be in any of the testing or anything like that, but I urge you to see some of the emergency procedures that can be done on the eyes, specifically a lateral canthotomy if you've never seen one. I wouldn't recommend that if you don't have a strong stomach, but if you're interested in that kind of thing, moving on in the medical field and emergency medicine in general, seeing a lateral canthotomy is pretty awesome. You can check out a YouTube video on that. All right, so talking about some of the tunics of the eyeball here. So the fibrous tunic, which is the outermost layer of the eye, provides mechanical support and serves as an attachment site for the extrinsic eye muscles and contains the cornea, which permits the passage of light and whose curvature aids in the focusing. And there's two things in that. You can have the sclera, which are dense fibrous connective tissues that form the white of the eye, and the cornea, which is the transparent anterior portion of the sclera. I also have the vascular tunic, which is the middle layer, also known as the uvea. And a couple structures associated with that. So the choroid, or a highly vascularized brown pigmented layer in the eye that prevents nutrients to all eye tunics. You can have the ciliary body, which is a thickened ring of tissue that kind of encircles the lens and is regulated by the ciliary muscle, which controls the lens shape by pulling on the spensitory ligaments that extend from the ciliary processes. We can have the lens, which is a biconvex, transparent, flexible disc that focuses light onto the sight receptors of the inner tunic. The lens is avascular, so you should keep that in mind. The iris, which is visible through the cornea, which is the colored portion of the eye, and contains blood vessels, pigmented cells called melanocytes, and layers of smooth muscle fibers. And when these muscle fibers contract, they can change the diameter of the pupil. So when few melanocytes exist, the eyes appear blue. Intermediate numbers, they're going to appear green. And when large number of melanocytes exist, they're going to appear brown or black. We also have the pupil, which is a round central opening that allows light to enter the eye. We also have the nervous tunic, or the retina, so the innermost portion of the eye. So the retina consists of a thin outer layer that absorbs light and a thick inner layer that contains photoreceptors. So those are cells that are sensitive to light. Some of the structures that are associated with that are cones, which are photoreceptors that operate in bright light and provide high acuity color vision. Rods, which are photoreceptors found in the periphery of the neural retina and distinguish vision in dim light and provide our peripheral vision. You have the macula lutina and the fovea centralis, which are lateral to the blind spot, and this is the region where light passes almost directly to the photoreceptor, which greatly enhances our visual acuity. And the fovea centralis is only cones, while the macula lutina is mostly cones. So from the macula lutina outward, cone density decreases and rod numbers increase. We also have bipolar neurons, which are rods and cones of the retina that synapse with bipolar neurons that depolarize and transmit the nerve impulses to the ganglion cells. Speaking on the ganglion cells, those form the innermost layers of the retina, and the axons of the ganglion cells converge to the optic disc to form the optic nerve. The optic disc is an area of the retina where there are no photoreceptors and is therefore commonly called the blind spot. And that's found at the base of the optic nerve that exits the back of the eye. We also talk about the inner chambers of the eye and some of the associated fluids there. So in the posterior cavity, which is segmented behind the lens and is filled with a gel-like substance called the vitreous humor, also known as the vitreous body. In the anterior cavity, which are the segments in front of the lens that are filled with a watery, nutrient-rich fluid called the aqueous humor. And that's subdivided into the anterior chamber between the cornea and the iris, and the posterior chamber between the iris and the lens. We also have the canal schlemm, It's an unusual passageway that encircles the eye and kind of drains aqueous humor into the venous blood supply. So, some of the accessory structures on the eye, we have the eyelids, which is the thin skin covered folds that's supported by the tarsal plates, which are connective tissue sheets that anchor the orbicularis oculi and the levator papillae superioris, uh, and those help to protect the eyeball. We have eyelashes, which are robust hairs that extend from the margin of each eyelid and help to shield sunlight, prevent foreign matter from getting in the eye, things like that. Um, you have the palpebral fissure, which is the eyelid slit that meet at the corners of the eye, called the medial canthus and the lateral canthus. And in the medial canthus, it's a fleshy lacrimal crunae. You have the eyebrows, which are short, coarse hairs that kind of overlie the supraorbital margins, and they help to shade the eye from sunlight and prevent perspiration from trickling down into the forehead. You have the tarsus gland, which is a modified sebaceous gland that produces an oily secretion that lubricates the eyelid and the eye to prevent the eyelids from sticking together. You have the conjunctiva, which is a transparent mucosous membrane that connects the eyelid to the eyeball and produces lubricating mucus to prevent the eye from drying out. And it's usually composed of stratified squamous epithelium, and an infection to this area is called conjunctivitis, or what we consider to be pink eye. We also have the lacrimal apparatus, which produces, distributes, and removes tears, and it consists of a few ancillary structures. So the lacrimal gland, which is an almond-shaped gland that is located lateral and superior to the eyeball, and that produces tears that are slightly alkaline and contain antibacterial enzymes called lysozymes and antibodies that attack some pathogens. We also have the tear ducts that deliver those tears from behind the lacrimal gland and to the space behind the upper eyelid. You have the lacrimal punctum. So those are two small pores on either side of the lacrimal peduncle, uh, and drain the tears from the eyes. We also have the lacrimal caniculi, which are small canals that connect the lacrimal puncta to the lacrimal sac. The lacrimal sac is a small chamber that nestles within the lacrimal sulcus of the orbital of the eye. And then you have the nasolacrimal duct, which originates at the inferior tip of the lacrimal sac, and it passes through the nasolacrimal canal to deliver tears to the nasal cavity. So the extrinsic eye muscles, you can see those all here. This just speaks a little bit about the neural tunic layer, which we covered when we were going over tunics. This just highlights a picture of it. And finally, a little bit more on the lens. Um, Again, a biconvex, transparent, flexible disc that focuses light onto the sight receptors of the inner tunic. Um, Again, remember the lens is avascular. So getting into some of the physiology of vision, we have refraction of light rays, so the bending of light by the anterior and posterior surface of the cornea, as well as refraction by the lens that cause light rays to come in and exact focus onto the retina. So the visual axis of the eye is an imaginary line drawn from the center of an object that you're looking directly at and through the center of the cornea and the center of the lens to the retina. Also, to form an image, you must have accommodation, so the lens must change shape in order to adjust for vision at various distances. So for close-up vision, the ciliary muscles contract and the lens is rounded and bulging, and the pupil constricts. So near the point of vision, you have minimal distance from the eye to an object that can be clearly focused on with maximum effort. Um, so for distant vision, however, the ciliary muscles relax and the lens flattens and the pupil dilates. So we also have far point vision, so distance beyond which there is no change in lens shape um, can increase focus. So usually that's about 20 feet. So the highest visual acuity is at the focus centralis. So once the light stimulates the rods and cones of the retina, nerve impulses are generated and sent to the brain via the optic nerve. So all images are inverted, so upside down and backwards. And visual pathways that lead to the brain. We have those photoreceptors, so the cones and rods that stimulate the bipolar neurons, which in turn stimulate the ganglion cells. The ganglion cells will then converge on the optic disc and penetrate the wall of the eye and proceed towards the diencephalon. as the optic nerve. And the two optic nerves, one from each eye, converge at the eye optic or the optic chiasm. And from that point, the impulse travels from the optic tracts to the brain. So approximately half of the fibers proceed to the same side of the brain, while the other half cross over to reach the opposite side of the brain. So the perception of visual images reflects the integration that occurs in the visual cortex that's located in the occipital lobe of the cerebrum. I lost our spot here. My apologies. Sometimes technology isn't what we always want it to be. Um, so I'm talking about central processing a little bit, and in the same regard, sensory pathways. So spinal cord and the brainstem. So information is processed on an ascending pathway from the sensory receptors to the spinal cord and the brainstem. And somatosensory pathways are divided into two separate systems. So we have the cranial nerves, which are receptors from the head and the neck, and the spinal nerves, which are receptors from the rest of the body. The dorsal column system, that begins with the axon of the dorsal root ganglion nerve that enters from the dorsal root, which joins the dorsal column of the white matter. And within that, you have the fasciculus gracilis, which contains axons from the legs and lower bodies, and the fasciculus cuneatus, which contains axons from the upper body and the arms. We also have the spinothalamic tract, which begins with a neurons in the dorsal root ganglion, and these nerves extend their axons to the dorsal horn, where they synapse with the second neuron, which is located in the thalamus in their respective pathway. You also have the diencephalon. Um, and within that, we have the hypothalamus, which is in charge of somatic and autonomic limbic systems. Um, we have the thalamus, which is an important relay for communication of the gateway between the cerebrum and the rest of the nervous system. And sensory input of the thalamus comes from most of the special senses and ascending somatosensory tracts. Each sensory system is related to a particular nucleus in the thalamus. And one of the exceptions to the rule is the olfactory system. So, olfactory tract axons from the olfactory bulb project directly to the cerebral cortex, along with the limbic system and the hypothalamus. So, thalamus is a collection of several nuclei that can be categorized into three anatomical groups. White matter that runs through the thalamus kind of defines the three major regions of that area. And lastly, in this, so just mapping of that retina visual cortex. Remember that visual field projects through the lens and falls on the retina as a reversed image.